Good afternoon, viewers. My name is Ferdy Venter, and this afternoon we are going to entertain ourselves with meiosis, which is a very important topic in the grade 12 life sciences curriculum, and many of the other content topics like reproduction and genetics has meiosis as a starting point. So let us immediately get involved. This afternoon's broadcast is suitable for teachers, for life sciences educators, as well as learners. Welcome to this broadcast, and I hope you will enjoy it. Let's go to the desk cam, and I've made us some very simple and large writings and drawings. So this afternoon we are going to play around with nice figures and drawings, and hopefully after today you will be able to understand meiosis a little bit better. Of course, when you look at a broadcast like this, it's always convenient to pause at any stage to maybe familiarize yourself with something that was said, so that you can have a look at a picture again or a diagram again and understand it much better. First of all, a quick reminder to tell you that, but just remember, a part of reproduction, and I will refer to it later, has also to do quite a lot with the topic of meiosis, and that is gametogenesis. Gametogenesis in reproduction is nothing else but meiosis, but the actual questions on meiosis itself will fall under paper 2. And if I'm not mistaken, it is 22 marks out of the 150 in paper 2 will contain questions on meiosis. Now, the first question you have to ask yourself is the 3 W's and maybe 1 H, WWH. And that WWH is abbreviations for where, why, and when does meiosis takes place. Where, why, and when, and the H, www.mindstream.co.za. When you go to that website, all the topics, grade 10, 11, and 12 life sciences, are shown there under the branch of life sciences, and especially under the subheading COVID-19 support. You will see a lot of material, past papers, glossaries, mind maps on different topics in life sciences. So enough of said about that. While I'm talking this afternoon, have a look at these terms, keep it in mind, and see if you, at the end of the day, if we have tackled at least all these topics, these terminology parts, with explanations on their part that they play in the process of meiosis. I'm not going to go through them now. Just familiarize yourself. Maybe there's a few other terms that's not even included here, but it's important that you are able to identify and define these terms. Back to my diagram, or rather my large, colorful writing, to make it as simple as possible. First question, where does meiosis takes place? When I'm saying where, I mean where exactly in animals and plants, in other words, in which organs, and then secondly, where in the cell does this process happen? Looking at animals, looking at mammals that we form part of, in the male animal, it happens in the testes, and of course, in the female animal, it happens in the ovaries. in the testes, and in the ovaries, the process of meiosis takes place for a particular reason. And in plants, it takes place in the ovule, and the ovule is also contained in the ovary of a plant. And of course, that is the female part of the plant, the ovule, or the ovules, and in the anthers. Now, I brought along a diagram on my cell phone that I'm going to slip in here. Quickly, I Googled meiosis in plants, and I got to this diagram, and I hope you can see. Just to make sure we understand the difference between an ovary and ovules. In Afrikaans, vir Afrikaanse kykers, sal julle onthou, leerders, lang terug in een van die vorige grade, het ons gepraat van die stempel, die styl en die vrugbeginsel in 'n plant. So in 'n vrugbeginsel kry jy saadknoppe. Nou dit is binne in daai saadknoppe waar meiose plaasvind. In English, in the ovaries, inside the ovaries you get numerous ovules, and inside those ovules you get the process of meiosis. So the ovary are the bottom larger part here with the smaller little circles inside the ovules. This is the female part. And the male part of the flower, of course, we call the anther. The anther. And inside the anthers, you will get the process of meiosis with the product then pollen grains. And those pollen grains are the product of meiosis to be distributed for further functioning, for fertilization, in other words. Right, so those are the locations in a plant and in an animal. I hope that makes sense on the where in plants and animals. And I start with these definitions, or rather locations, to make sure that learners understand the bigger picture of meiosis. Then secondly, in a cell, and I'm going to show you lots of diagrams this afternoon to show you exactly where in the cell does the different phases, because it happens in different stages of the cell. First of all, it starts in the nucleus, and I will show you later where this nucleus is. And then, as meiosis progresses, the nuclear membrane at a stage disappears, and then this meiosis happens all over in the whole cell, animal as well as plant cells. Very important. Meiosis happens in animal and plant cells. Secondly, why does meiosis take place? Why is it necessary for meiosis? And remember, in grade 10, you have done mitosis, the cell division process where happening in somatic cells, in body cells, to multiply, to have the same number of chromosomes, and the function of mitosis, if you remember correctly, was for growth and for the recovery of maybe damaged cells, and also in very simple organisms, simple animal organisms as well as plant organisms, by binary fission, you can also get the process of mitosis, which is then part of reproduction. So that's mitosis. But meiosis purpose is to form gametes, male gametes as well as female gametes. And these gametes must be haploid. In brackets we write a single small letter N. Diploid we write with a 2 in front. 2N is diploid. So meiosis is the process where diploid cells change or divide to form haploid gametes. Now, give me some examples of gametes. Male gametes. In the animal, they are sperm. In the female, they are the ova. Plant or animal, as I already said. So this is the first reason why meiosis is necessary, to form haploid gametes in plant and animals. Secondly, because meiosis is called reduction division, it means that the chromosome number is divided in half. Because after fertilization, cells with half of the chromosomes of that particular organism are fusing with cells also with half to give you the diploid number. Let me take an example. In humans, a male or a female gamete has a chromosome number of 23. In other words, 23 chromosomes in each and every cell of that organism. When fertilization takes place, in other words, when a male gamete and a female gamete fuses, the product is a zygote with 46 chromosomes. In other words, there was a doubling effect of chromosomes. Now, meiosis is responsible for halving the doubling effect of fertilization to reduce a diploid number into a haploid number. And remember, each and every organism has its particular chromosome number. Humans, 46 chromosomes. Other organisms, 48 chromosomes. 8 chromosomes, 22 chromosomes. It differs from species to species. And the last and also very important purpose of meiosis is to provide genetic variation, to lead to genetic variation. And that you will understand when we come to the process itself. When does meiosis take place? It can take place at any stage in a plant or an animal organism, but that organism must be mature. It must be an animal or a plant that has reached maturity. And after maturity, any animal and any plant which is fertile, of course, can undergo meiosis. Now, with that in mind, let us quickly look at the difference between what you already know of mitosis and what we are now doing today, meiosis. And it doesn't matter if schools in this time of COVID are a bit behind with the annual teaching plan and maybe haven't started yet with meiosis, then this is pre-knowledge to help you understand when the teacher in class explains it. If you have done it already at school, now you can start doing revision on this process. Quickly, mitosis is the division of a parent cell after DNA replication, first of all, into one cell. One parent cell divides to form two daughter cells, with the chromosome number staying exactly the same. The chromosome number there, in that nucleus, is 4. After the first part, the replication part, it is again 4. And after the process of mitosis happens here. It happens right here. The product is from one cell, 2 cells, still with 4 chromosomes in each daughter cell. In other words, the chromosome number stays the same. Because when there's regeneration of cells, when there's growth, when there was an injury and cell division is responsible for healing a particular wound, the chromosome number of that somatic cells must stay the same, depending on the animal or the plant where it happens. Now the difference, looking at the opposite diagram of meiosis parent cell also undergo DNA replication. Then we have two divisions, a meiosis I and a meiosis II. After meiosis I, two daughter cells, and look what happened. The 4 chromosomes, it also started with 4 chromosomes. And that 4 chromosomes was reduced now to 2 chromosomes in each daughter cell. And from there, there's a second meiotic division, and the product of each of that divisions is daughter cells with, again, 2 chromosomes that just grow to maturity. So, meiosis, one cell divides through 2 meiotic divisions into 4 new cells, each containing half the number of chromosomes it started with. So each of these cells has 2 chromosomes inside. Also this one. That is the difference between mitosis and meiosis, looking at it from a bird's eye view. Now with that in mind, let us quickly refer you to these 2 processes that you will do in one of the later topics, and that is under the topic of reproduction. You will learn about spermatogenesis and oogenesis. Spermatogenesis, okay, the 2 together is called gametogenesis, the formation of gametes. Spermatogenesis, of course, in males, and oogenesis in females. Let's write it there on top. In males, and oogenesis in females. Now, I'm not going to concentrate on this process this afternoon because it's part of reproduction, but keep in mind, this is nothing else but meiosis. And it shows you also a first mitotic division, a first meiotic division after mitosis, and then a second meiotic division. And in the case of spermatogenesis, the results are sperm. Each and every cell that undergoes meiosis in a male forms a haploid sperm cell. And each of these sperm cells are able to fertilize an ovum. But in a female, it produces also 4 cells at the end, of which 3 degenerates to provide an ovum or an egg cell, but please let us refer to an ovum, which of course again is haploid. The sperm cells are also haploid. Now let's take again the example of humans. In a human, for the demonstration here, there's only 1, 2, 3, 4 chromosomes. The product, each gamete, has 2 chromosomes. So this is some other organism. If it was in a human, there would be 46 of those little chromosomes, the same there. And after meiosis, the product would be 23 chromosomes in each sperm cell and 23 chromosomes in that ovum. Now you understand the halving effect of meiosis, so that a sperm cell with half of the set of chromosomes, in other words haploid, can fertilize a haploid ovum, and the product will then be again a diploid zygote, the product of 2 gametes that has fused. All right. So with this in mind, this as background, this is still part of the purpose, the function of meiosis. Let us now go and look at the process itself. Now I'm going to try, my diagrams are very large, so hopefully you will be able to see the big picture, and I'm going to move these diagrams around so that you can clearly understand. So now we come to the how of meiosis. And before I do that, remember we refer to the set of chromosomes in each and every organism. When you look at a diagram, a set of diagrams or a diagram of all the chromosomes in an organism, we call it a karyotype. I did not bring along a diagram of a karyotype. Go and refer to your textbooks. But the karyotype shows you exactly the combination and the organization and the arrangement of chromosomes inside each and every cell of each and every organism. Again, humans will have a set of 46 chromosomes. And let me just mention it. Of that 46, 44 pairs, 22 pairs, in other words, 44 cells will be called the two of them will be called the gonosomes, and the 44 would be called the autosomes. Autosomes 44 plus 2 gonosomes gives you a set of 46 chromosomes. And that autosomes are responsible for bodily characteristics, and the 2 gonosomes for sexual characteristics, amongst others, determining male and female characteristics. But we are not going to stand still at that this afternoon. Now the first phase, let me be very clear. The first phase I'm going to tell you about is the interphase. But please keep in mind, the interphase of meiosis is actually, I'm immediately falling into the trap that I'm trying to explain. The interphase is not actually part of the process of meiosis. It is a phase that happens in between 2 consecutive meiotic divisions. Now, I've drawn a cell here clearly to try to explain to you, and remember, this is not a complete cell. I've drawn a cell that clearly shows the cell membrane, in this case. If it was a plant cell, there would be a cell wall also right around this cell on the outside. There would be a cell wall as well as a cell membrane. Then it would be a plant cell. So this is an animal cell with only a cell membrane. Then inside you'll have the cytoplasm and organelles that I did not draw in this diagram, like chloroplasts, mitochondria, Golgi apparatus, ERs, vacuoles, lysosomes, etc. I did not draw them for the obvious reason to simplify the diagram, to concentrate on meiosis. But keep in mind, they are there. What we are going to concentrate on is the nucleus of a cell. Now, in the state of interphase, the nucleus consists of a network of chromatin. We call it also a chromatin network. In other words, chromosomes are not visible as such, but male and female chromosome material are in this set or this grouping of chromosome material, or then DNA material. And for our explanation, I used green and red. And let us assume for this afternoon's explanation that green represents female material and red male material. In other words, the red material comes from the father, and we call it the parental genetic material. And then the female material comes from the mother. In other words, an organism to which this cell belongs to. Let's say this cell belonged to me, to Ferdi Venter. Then the green material, DNA material, I got from my mother. And the red material I got from my father. All in combination in each and every cell in my body inside the nucleus. I drew this nucleus to show you that a nucleus consists of a nuclear membrane, which is a double membrane, with nucleopores. Many times in our diagrams in grade 12, they will only draw one line here. They will draw a solid line to show the nuclear membrane. But keep in mind, it's a double membrane under a very large enlargement. This nucleus also has a nucleolus. Kom ons sê dit net gou in Afrikaans. 'n Kernmembraan, kernporee en 'n nucleolus, kernliggaampie. And then of course, the nucleoplasm, die nucleoplasma, of dan nou kernplasma. So in this state, the cell is in the interface. Now, what is the cell doing during this phase, which is in between two meiotic divisions? The cell does its normal function. If it is a brain cell, then the cell will do, it will be probably neurons. This will be part of a neuron, and it will have to do with brain functions, these cells. If it's a blood cell, an erythrocyte or a leukocyte, it will does the purpose of carrying oxygen or carbon dioxide. If it's a skin cell, an epidermal cell, or an epithelial cell, or a connective tissue cell, it will do that particular function. It will perform that particular function. But also in this interface, a very important process happens, and that is replication. DNA replication. Now you have studied that in the part of DNA in the section just before this chapter or part of the work of meiosis, and that is replication. What is replication? Replication is the doubling of the genetic material of these threads, which are not visible yet as separate chromosomes. It's a pity we are not able to show each and every one of us, go to an electron microscope and see these things under the microscope. Maybe if some of you go and study this part of the work, you will be fortunate enough to have a look at that, because it's a beautiful and a wonderful process. So replication in the interface, as well as the cell doing its normal function. Let's move on. I'm still showing you the interface. I'm still showing you the interface. Same cell. Don't worry if it doesn't look the same as the previous diagram. Remember, I'm also human, and I'm trying to make the cells look exactly the same to show you it's the same cell that I'm talking about the whole time. So let us play with my explanation. Now, what I did, I've shown this chromatin network, so-called falling apart and become visible as chromosomes actually in the next phase. But I want to demonstrate the process of replication in these two chromosomes. So if you get a diagram like this with a label, this will be a chromosome, and again in red, it represents chromosome from the father, a paternal chromosome. So this chromosome I inherited from my father, and this chromosome I've inherited from my mother. So my chromosome set, I'm a human, so I will have 46 of these guys. 23 from my father, 23 from my mother. to form a full diploid set of chromosomes. So in the interphase, replication happens. I've only drawn 2 of those 46 chromosomes for the purpose of the explanation. Replication happens. You've studied the process of replication. I'm not going to repeat that. And this single strand or thread, rather a single strand. Remember, what is this? A chromosome is actually a DNA molecule. So there's a double helix arranged with, of course, genetic material, sections, nucleotides, as you've studied DNA. Each nucleotide or part of a DNA molecule represent a gene. Remember the word, a gene. And that gene has a particular characteristic in a particular organism. Eye color, hair type, hair color, whatever. Physical as well as other characteristics in the body. The same with the maternal chromosome. It contains chromosomes. Now, where are we moving towards? I've only drawn 2, so you can clearly see what's coming. These 2 chromosomes will soon become a homologous chromosome pair. At this stage, they are only separate chromosomes, but they are not actually visible as is in the interphase. I've just drawn it to show you the process of replication. After replication, look what happened. Look what happened. Let me put it in the order of how it happens. So after replication in the interphase, this chromosome, this single-stranded chromosome, becomes double-stranded or double in the genetic material. In other words, a single chromosome becomes a chromosome consisting of 2 chromatids joined by a centromere, still with the genes visible or appearing in that chromosome. So this gene here is now represented by a gene in each of these chromosomes. So if that gene represents eye color, then let's say that part and that part also represents eye color. And so for all of this genes, and remember the whole set of genes, all the characteristics in a human body, you will later learn in genetics, is called the genome of that particular organism. The same in the maternal chromosome. This chromosome is also called unreplicated. And that chromosome is called a replicated chromosome because it is after the process of replication. Now, as I said, this represents then the end of the interphase. The chromatin network, inside that, chromosomes undergo replication, not really visible like this. Let me be very clear. Not really visible like this, but for the purpose of the explanation, I've shown it like that. Remember, there are different phases in meiosis. Your teacher would have told you, you remember it by the abbreviation IPMAT. Ne? IPMAT. To remember it as interphase, then prophase, metaphase, anaphase, telophase. And in meiosis, there's a meiosis 1 and a meiosis 2. In other words, you have interphase, the phase between two cell divisions. You have a prophase 1, then a metaphase 1, an anaphase 1, and a telophase 1. After that, a prophase 2, a metaphase 2, an anaphase 2, and a telophase 2. Which happens one after the other, first meiosis 1 and then meiosis 2. Now, let's go to the first phase, and that is prophase 1. Same cell that we've started off with. And what happens now, and this is now important, in a test or an exam, it will be expected of you to identify a particular phase with reasons. Or you will be asked, explain what happened in each phase, or explain the behavior of the chromosomes in each phase. Now, as I said, in prophase 1, the first thing that happens normally is that the chromosomes becomes a little bit shorter and they become visible as two chromatids joined by a centromere. And again, I've drawn only one paternal chromosome and one maternal chromosome, called 1 and 2 in this case. I would have rather written here male and female. Remember what this is? This part comes from my father's genetic material and this part from my mother. I have 23 pairs, 23 of them coming from father and 23 of them coming from mother, giving me a full set of 46 chromosomes, in other words, a diploid set in my somatic cells. It's only in gametes that there's a single set, a haploid set, you will see later. Right, so now an important term must be mentioned. When they arrange like this, remember in the previous stage, they were just randomly, in the interface, they appear just randomly, even when they have underwent replication. They still are just lying randomly around. They are not arranged in any form whatsoever. But then part of the prophase 1 is that they arrange in so-called homologous pairs. Homoloë chromosoompare in Afrikaans. Now, what is the definition of a homologous chromosome pair? They have the same structure, more or less exactly the same size. They have the same characteristics that they represent. If it's eye color, hair color, and length of the individual, then you will have the same characteristics on the female part, the female chromosome, coming from the female mother, the mother. Then, and the centromere is exactly in the same position. Remember, this centromere can sit lower, it can sit higher, it can sit at any place here, any position rather. But where it is situated, it will be situated in the same location as on the other half of the arrangement. The place where these genes, and after replication, these genes are represented by so-called alleles. You will learn later in genetics, where an allele represents a particular characteristic. Let's take seed color. Then in half of that genetic material, in other words, in the paternal part, it can have, let's say, a green seed, and in the female part, it can represent white seed in color, and then it is called heterozygous. But you will come to that. Don't worry now about that. But the positions on the chromosome where these alleles are situated are called the loci. Singular form locus, plural form loci. You will learn more about that later on. Still prophase. Chromosomes are visible, remember, as replicated structures. Product, it's the product of replication. When they were single, they were called unreplicated chromosomes. These replicated chromosomes are visible as chromatids. There's a chromatid, there's another chromatid, there's the centromere. There's a chromatid, there's a chromatid, that is a chromatid, and this is the centromere. Now, still in prophase 1, a very interesting process happens. Now, a very particular process is happening where these structures come about. And this whole, if I can demonstrate it, this whole arrangement of threads is called the spindle, because it needs to happen, because part of prophase 1, the nuclear membrane and the nucleolus has disappeared. It disappeared. So that these structures are now, can I say, all over this cell. There's nothing keeping them in place except this spindle. And a spindle consists of spindle fibers, literally little threads, draadjies in Afrikaans, spoeldrade of spoelvesels, coming from what we call the centriole. Don't get confused with a centriole and a centromere. Centromere keeps two chromatids together. Centriole is an organelle in the cell. The position or the environment in the cell where the centriole comes about is called the centrosome. Normally it's very difficult. In an exam you will get a label, and currently we are marking correct centriole or centrosome. But the difference is the centrosome is the area where the centriole, the organelle, develops. And the centriole gives rise to spindle threads. And the spindle threads keep these chromosomes in place. Now, in this diagram, there's only one pair drawn. In humans, there would be 23 such pairs. In other words, they will go up there, and where do they arrange? Not yet. It's still prophase, so they could even be not organized here exactly in the center, because this is actually the next part of this process. I've jumped the gun a bit with this diagram, because this is still prophase. So it doesn't mean they are lying on this center region, which is called the equator of the cell. The Earth has an equator. A cell also has an equator. But just keep in mind, part of prophase is this process. And we call this process where a part of this leg or this chromatid overlaps with the female part. The process where it happens is called the chiasma. Singular form, chiasma. Chiasma on one pair. Another chiasma gives you chiasmata. Or if crossing over, which is the process's name, happens there, and maybe also a little bit downwards in the same pair, these two positions will be called chiasmata. And then something strange happens here. Those two threads, or chromatids rather, let's call them chromatids, they break, literally, and they share. The red or male part, the male chromosome, receives a little bit, a piece of the female part and vice versa. So the red one is still male and the green one is still female material, chromosome. And then after this, the spindle is formed. Crossing over has taken place. These threads have formed the spindle, as I already said. A chromosome pair is formed, introduces or represents the end of prophase 1. Just one more term. This grouping, let me go to the previous diagram. If you have a label in the exam showing you both of these chromosomes, they are not involved in crossing over yet. Then they are called a homologous chromosome pair. So you will get 23 in a human, 23 of these homologous chromosome pairs. Only one is drawn here. The minute they became involved in the process of crossing over, this homologous pair is called a bivalent. Bivalent means a homologous pair involved or part, busy with the process of crossing over. It's a bit of technicality, it's a bit of a technical point, but keep that in mind when you study this work. That is the end of prophase. Take notice, no nuclear membrane, no nucleolus. So this set of chromosomes is held in place by a spindle. So the spindle connects all these chromosome pairs, and normally they are held. Remember, these threads can go be at the back or above. In a three-dimensional vision, it forms a bundle or a group or a spindle, as I said, and normally it is fixed. It is fastened to the centromer. Later you will see when it shortens what happens. Right, end of prophase 1. Metaphase 1. As I said, I jumped the gun a little bit with the previous diagram. Those two chromosomes in that pair could have lain in any way, could have lain any way, any arrangement. But in metaphase 1, they arrange on the equator. So those pairs will all lie on this equator and form part of this spindle. And the minute you see this arrangement, in other words, crossing over is completed. This paternal chromosome has a little bit of the maternal chromosome, and the other one a little bit of the male chromosome. I haven't shown. You see there crossing over takes place there as well as here, but I've drawn only the top part. Forgive me for that. I haven't shown the interchange of DNA material at the bottom to make it just a little bit simpler. Metaphase 1 is a very short-lived phase, and the only thing that happens, chromosomes arrange on the equator. The spindle extends across the whole cell. Each chromosome lies on equal sides of the equator, and the centromers attach to the spindles and the spindle fibers. Metaphase 1. Telophase 1. No, not telophase 1. Anaphase 1. The spindle fibers shorten and pull each chromosome of each chromosome pair to opposite poles of the cell. Spindle fibers shorten and pulls this chromosome towards that side, and those spindle fiber pulls these or this chromosome to the other side, and this happens to all let's say 23 pairs or 46 chromosomes in this cell. And that is all that happens in anaphase 1. And then you get telophase 1, where these chromosomes that has moved to the poles reaches the poles, and each of the poles now has half of the number of chromosomes of the original cell. In other words, reduction division already took place. The cell membrane constricts and divides the cytoplasm in half to form two cells. Sometimes this does not even happen, but many times it divides, it forms two technical, visible two cells, and then meiosis II starts. Each pole of the cell forms then a new. And sometimes the nuclear membrane and nucleolus forms again, but sometimes not. It goes straight from this phase to meiosis II without even forming a new nuclear membrane and nucleolus. Right, end of meiosis I. Meiosis II, remember, it should now be a continuation from telophase 1 to prophase 2. As I said, two separate cells can be visible or not. They can be divided fully with then half of the chromosomes. Remember, we started with 2 chromosomes. Now there's 1 chromosome, full chromosome, consisting of 2 chromatids and a centromere in each of these new cells. The little spindle has either disappeared fully or a little bit and now start forming again, because it has to pull again something towards the poles. Prophase 2. Remember to write this 2 in the form of a Roman 2 or a normal figure 2 every time when you write down a phase. Otherwise, you won't get your mark. So from there, prophase 2, metaphase 2 must happen. What happened in meiosis I? In metaphase 1. The homologous chromosome pairs arranged on the equator of the cell. In prophase 2, single chromosomes arrange on the equator of the 2 new cells. Again, only 1 is drawn. Again, in humans, there would be 23 of these chromosomes on the equator. Paternal chromosomes, with a piece of the maternal chromosome still there, depending on how many overlaps and crossing overs took place. Same there, maternal chromosome with a little bit of the father's genetic material. And this, can you clearly see, is where genetic variation will happen in the next generation. This is metaphase 2. Anaphase 2. Anaphase 2. What happened in anaphase 1? Spindle fibers contracted and pull full chromosomes of the chromosome pair towards the poles of the cell. But in anaphase 2, something happened. The centromere splits in half. Centromere splits in half. And so-called daughter chromosomes or single chromatids are drawn in this case towards the poles. In other words, this was part of the original chromosome, paternal chromosome, and that part of the maternal chromosome pulled towards the poles. This spindle still, I just didn't draw it, but still are still formed to keep these things in place inside the cytoplasm. And then lastly, telophase 2. Telophase 2, which is the last phase of our process of meiosis. Each of these cells split in half. The process is called cytokinesis, where there's a split in the cytoplasm, cytokinesis, to form then, from one of these cells, again, 2 new cells. In each, a new nuclear membrane develops, as well as a new nucleolus in each of these newly developed cells. A full nuclear membrane developed after this. Now, let's look at the arrangement or the behavior or the appearance of chromosomes. Remember, we started with 2 chromosomes, prophase 1. There were 2 chromosomes. Where is my prophase 1? Prophase 1, 2 chromosomes, 1 and 2, paternal, maternal. Product of meiosis. From one cell, 1, 2, 3, 4 cells, each consisting of, in this case, 1 chromosome, and there's a little piece of the maternal material, and there's still the piece of paternal. And it looks like there's no genetic variation, but there is. I just didn't draw it to simplify our discussion. One cell with half the set of chromosomes. So these cells are now haploid. The initial prophase cells, that one was diploid, double set, full set of chromosomes. Product of meiosis, haploid cells, 1N. In a male or in the plant cell, when pollen is formed, in a male animal, each of these cells develop a little tail, and then it is called a sperm cell. Each of them can form a sperm cell. So if there's 1 million cells, mother cells at the beginning, there will be 4 million sperm cells formed from that 1 million. 4 times, 4 new chromosomes, cells from one initial mother cell. Telophase 2. Can you now see the purpose, the significance of meiosis? In the beginning we said, to form haploid gametes, you saw it happen. To entertain the halving effect to bring about the halving effect after the doubling effect of fertilization. Here you see it. The diploid set is divided into a haploid set. And to introduce genetic variation. Because if this cell is now one sperm and it fuses with an ovum, then in the new individual, there will be genetic material of the grandmother or father of this new individual. Remember, this is, let's say, my cells. This is part of my mother's material. My children will have, in other words, a little bit or part of my mother and my father's material. In other words, they will have material from their grandmother or grandfather, 2 generations back. And that is why a particular characteristic can be hidden in 1 or 2 or 3 generations and then suddenly appears in a 3rd or a 4th or a 5th generation. It also happens in genetic disorders. This exact characteristics of not being visible or coming about in a particular generation. You will learn about it in genetics. Right. So I think this enough of the phases of meiosis. Now, something strange sometimes happens in the process of meiosis, and that is called abnormal meiosis. And we are nearing the end of this presentation. Also a very popular question in grade 12 life sciences papers. Abnormal meiosis. And very important, abnormal meiosis can happen in meiosis 1, there, or in meiosis 2. Let me write it somewhere, there on the right-hand side. This process here is meiosis 1. I like the Roman one. And this part here is meiosis 2. So this process that is called non-disjunction, very importantly, can happen in meiosis after meiosis 1 or after meiosis 2. Now, what happens is there is an absence of division of chromosomes, of splitting of chromosomes, or of daughter chromosomes. Let's first take the scenario, it happens in meiosis 1. That is normal meiosis. Can you see? Initial mother cell with 2 chromosomes. And look what I said, a homologous pair of chromosomes will must have the same characteristics and appearance. So there's 2 chromosomes forming, the same as my diagrams earlier, forming one homologous pair of chromosomes. Meiosis 1 happens and the product is 1 chromosome in each new cell, reduction division. In Afrikaans reduksieverdeling. 'n Stel van 2 kromosome verminder met die helfte en word 1 kromosoom in elke sel. Normale meiose. But here, abnormal meiosis happen. Instead of those 2 chromosomes, part of a homologous pair, 1 chromosome moved to each side, one side of the cell, and the other chromosome to another side. Both chromosomes move to the same side. In other words, the spindle threads pulled both chromosomes to one side, one cell. And the other one receives nothing of this pair. Remember, in a human, there's still 22 other pairs. So the other 20 pairs can happen normally, like there. But then this one pair has 1 less chromosome on this side, but 1 extra chromosome on that side. And this process is called non-disjunction. Nie disjunksie in Afrikaans. Nie verdeling. Let's finish with this part. Then meiosis 2 happens. Those 2 chromosomes split. Remember, centromere splits, and a daughter chromosome or a chromatid moves to the one side and form a new cell, again with 2 chromosomes. From 2, it still stays 2. Reduction has already taken place. And there's now, in each of these new cells, there's now half the set of chromosomes. In other words, this one has now an extra chromosome. If you go there, there's a normal gamete from this scenario. There's a normal gamete with 1 chromosome. But there's an abnormal gamete with 2 chromosomes. And there's an abnormal gamete with 0 chromosomes of that particular pair. Again, I say, there could be other chromosomes from other pairs, but in this pair, 0. And this happens, okay, let me not jump the gun. That's a normal one, abnormal. But when it happens in meiosis 2, when this chromatid, or when this centromere divides, same story. 2 chromatids move to one cell, and nothing to the other cell. This is the normal process. 1 chromatid to the one side, 1 chromatid to the other side, 2 normal gametes. Male or female, doesn't matter. And then this non-disjunction happened in meiosis II. Now, in humans, this is how Down syndrome comes about. Down syndrome, a genetic disorder where there are certain characteristics visible in an individual because of an extra chromosome that take place, that happens, that appear in chromosome pair 21 of the karyotype. And that is why it is called also trisomy 21. It happens on chromosome pair 21, this extra chromosome. So a gamete comes about in humans with 24 chromosomes instead of 23. One extra. And that one extra gamete fuses with a normal gamete to form then a new individual where the zygote has 47 chromosomes. And the embryo will have 47 chromosomes in each cell. And the newborn baby will have 47 chromosomes in each cell. And all those extra characteristics that a Down syndrome person has is because of this extra chromosome from chromosome pair 21. Same there. If that gamete with an extra chromosome fuses, not with this one because this is from the same individual, from another individual with a normal set like this, an extra chromosome appears at position 21 and it leads to Down syndrome. So make sure, learners, that you understand the process of non-disjunction and then how Down syndrome comes about. I'm finishing off. Remember, we have discussed now literally all these terminology, all these terms. If I quickly scan my list, we really referred to all of them. As I earlier said, this material is available on that website, Mindstream, but we also provided it to our teachers, so you can very easily ask your teacher if they can maybe print you a little booklet of this material. This is the first page of that particular material. We've discussed where and why and how. Another popular question could be the difference between meiosis and mitosis. And I'm not going to go through all of this. I'm going to leave it there, visible, so you can pause. You can take a picture with your other device. If you're looking on a cell phone, use your tablet and take a picture or vice versa. And there you see a list of differences between meiosis and mitosis. Not less than 1, 2, 3, 4, 5, 6, 7, 8, 9. Normally there are 2 or 3. When you draw a table, you will immediately get 1 mark, and then 1 mark each. So if there are two differences, it will count 5 marks. 1 for the table and 1 each for the differences. So there are some differences between the two processes. And then also, what are the differences between meiosis 1 and meiosis 2? Remember, meiosis 2 is nothing else but mitosis. That happens after meiosis 1. Crossing over, no crossing over. Chromosomes arranged at the equator in homologous pairs. Chromosomes arranged singly at the equator in metaphase 2. Homologous chromosomes pairs separate. They call it sister chromatids in this case. Or they call it daughter chromosomes, or then just chromatids separate. Whole chromosomes are pulled to opposite poles. Chromatids are pulled to opposite poles. 2 diploid daughter cells are produced. 4 haploid daughter cells are produced. So please make sure you are able to identify some differences. Now, this is a typical answer in an exam paper. This is what this material is all about. It shows you typical answers. Importance, we have spoken about this. So there you will have a mark. Production of haploid gametes. The halving effect of meiosis overcomes the doubling effect of fertilization. There will be a second mark. In other words, to maintain a constant chromosome number. It is a mechanism to introduce genetic variation through crossing over, and this other one I did not mention, as well as the random arrangement of chromosomes on the equator. Let me quickly explain. If I can get my metaphase 1, metaphase 1 as well as metaphase 2. Random arrangement means the following. If chromosomes arrange on the equator, they can arrange like this, as shown in the diagram. Now let me use my fingers to represent the chromosomes. They can arrange like this. Randomly, they can also arrange like that, just flipped over. Now I'm going to disjoint myself, but they can also arrange like that. I tried to explain that. So they can arrange in any possible way, which is called random arrangement. Now, depending on the arrangement, you can clearly see if it's like this, that particular chromosome is on that side and there. But if you swap it around, the chromatid that has overlapped is now on the other side, so it becomes part of a new gamete, and that produces also genetic variation. It can also happen in meiosis too, where that single set of chromosomes arrange randomly on the equator. So that is the purpose. I've already referred to that terminology, the phases we have, and then I've already referred you to the process of non-disjunction and the purpose of that. Now, if we can come back to the camera, Mr. Cameraman. This brings us to the end of this presentation. I hope that in this hour that we have talked to one another, that I've showed you some practical explanations on the process of meiosis. Please use it. Please go and study it. Use your textbook, use your study guides together with it, and make sure you understand the different roles, the phases, and the importance of meiosis in plants and in animals. Thank you very much for being with us. Good luck for the rest of the year, and we see one another soon. Bye-bye.